Hi, hope you can hear me. So I welcome you all to the second consecutive webinar organized by the Human Genetics Society. Today our speaker is Dr. Heshan Peiris. I have known Dr. Heshan Peiris for a very long time, so it's an absolute pleasure to introduce him. He is a principal scientist at Tusk Inc., a drug discovery company which develops novel therapeutics for treatment of KRAS-dependent cancers and metabolic disorders. So prior to his current position, he was a research fellow at Stanford University, where he pioneered novel genetic screening platforms to characterize the role of type 2 diabetes risk genes identified by GWAS. His work in the functional genomic space has led him to develop new genetic screening platforms, organoid culture systems, and genetic perturbation models. And also he was awarded fellowships from the American Diabetic Association and Stanford Child Health Research Institute. And his work has been widely published in Nature Communications, PLOS Genetics, Cell Metabolism, and Diabetes. To start the webinar, I would kindly request all of you to keep your mics muted. And if you have any questions, there will be a Q&A session at the end. So I hand over this platform to Dr. Heshan now.
Thank you very much, Dr. Dineshini. I'd like to begin by thanking the Human Genetics Society for the invitation to present today, Dr. Dineshini and Dr. Nirmala for the invitation as well, and then for organizing this webinar. So today I'll be talking to you about a few platforms that I established with the aim of trying to characterize the role of GWAS candidate genes, particularly in trying to understand the role that they play in the pathogenesis of type 2 diabetes. Before we begin, I just wanted to give you a little bit of background about myself. After finishing my schooling at St. Thomas's and my undergrad at Manipal University in 2006, I moved to the Flinders University of South Australia, where I did my PhD, followed by a postdoc. After that, I moved to Stanford University as a research fellow in genetics in the Department of Developmental Biology. And then for about a year and a half now, I've been a principal scientist at Tusk, which is a drug discovery company. The main role there of myself and my team is to develop novel therapeutics, small molecules essentially, for the treatment of genetically driven and in most cases KRAS-dependent cancers. So we're talking about cancers in the colon, in the pancreas, and in the lung to some degree. So today I'm going to focus my talk about the work that I did in the functional genomic space while I was a research fellow at Stanford University. So in the first part of the talk, I'll focus very much on my work with genetic screens that I established. Firstly, in the Drosophila fruit fly, essentially trying to move from GWAS candidate genes that are identified by population genetics and really trying to understand how risk alleles contribute to the pathogenesis of a disease. I also talked to you about a human three-dimensional organoid culture system that I established that allowed myself and then subsequently the field to do genetic manipulations from tissues isolated from organ donors, which really allowed us to understand how a gene affects the function of pancreatic cells, and then more broadly speaking, how that feeds into the pathogenesis of metabolic disorders, and particularly type 2 diabetes. And then in the second half, I'll switch gears a little bit and talk to you more from the aspect of targeting genetic variants. So very much more drug discovery. How can we target genetic variants? I'll use the example of one type 2 diabetes risk gene and the small molecule screening platforms that I established, which really identified a family of very novel therapeutics and a new angle of treating metabolic disorders, especially beta cell dysfunction. So given that we'll be talking today a lot about type 2 diabetes, I thought I'd start with this very broad overview of glucose homeostasis. So by definition, glucose homeostasis is the maintenance of blood glucose levels within a physiologically acceptable range. What happens, however, when we ingest a meal that's rich in carbohydrates or has a high glucose content is that we really tend to push our blood glucose levels beyond that physiologically acceptable range. And what happens then in healthy individuals is that it triggers the release of insulin from a very specific group of cells referred to as the pancreatic beta cells. And once insulin is in circulation, it has a number of effects on peripheral tissue. In the skeletal muscle, it promotes glucose uptake from the blood via GLUT4 and well-documented transcriptional pathways. And then in the liver, it stimulates glycogen formation. The net effect of these two predominant pathways is to really reduce blood glucose levels and then clear it out of the circulation and bring it back to that physiologically acceptable range. So you can then appreciate that insulin, and more specifically, the beta cells in the pancreas that produce and secrete insulin, are critical to maintaining glucose homeostasis. So if we then look at the pancreas with a little bit more granularity, it is a very unique organ in that it is both exocrine and endocrine in nature. Now, the endocrine fractions of the pancreas are organized and dispersed throughout the body of the organ in the form of these pancreatic islets. Each one of these islets consists of approximately 1,000 cells, the predominant of which are the beta cells, which is the body's sole source of the hormone insulin. There's also a small fraction of these alpha cells marked in green in this figure, which produce and secrete glucagon, the hormone that counteracts the activity of insulin. So in a schematic of a beta cell, this is a highly specialized cell type. The glucose transporters on the surface of the cell allow glucose to be taken up. It is then metabolized, resulting in the production of ATP. And that ATP causes the closure of ATP-sensitive potassium channels, which causes the cell to depolarize. The entry of calcium subsequently triggers the exocytosis of insulin. So this is what we typically refer to as glucose-stimulated insulin secretion. It is the primary function of the beta cell, and there's many experimental ways to visualize this phenomenon. One example that I'll show you today on this slide and throughout this talk is what you can do in vivo with mouse models. So in this particular experiment, I took a cohort of mice, I fasted them overnight, and then the next morning, I injected glucose intraperitoneally, and then I followed a serum insulin levels in these mice for the next 2 hours. And then what you can observe very clearly here is that following a glucose injection, there is a rapid increase in insulin over the first 15 minutes, followed by a gradual decline over the next 90 to 100 minutes, back down to that basal level. So this is one way of visualizing glucose-stimulated insulin secretion in vivo. And this is what you can also replicate in patients in the clinic by orally challenging a patient with a glucose drink and then following insulin over the subsequent few hours. Conversely, what you see in type 2 diabetic patients is a loss of this ability to respond to glucose. By secreting insulin in response to glucose. So if we talk about type 2 diabetes, then it is a very common and chronic metabolic disorder prevalent in most countries throughout the world. And for many years, it has been postulated that the primary driver of type 2 diabetes, of course, is insulin resistance. And this is, of course, due to high BMI, increased circulatory fatty acids, etc. But more recently has been proven, though, that there's also a very strong genetic predisposition. There's a strong genetic link to an individual developing type 2 diabetes. And then with the advent of genome-wide association studies and RNA sequencing, which has become cheaper over the last 10 to 15 years, this has really been fine-tuned further to identify approximately 400 genomic regions that are robustly associated with type 2 diabetes risk. So to put this very simply, what this means is that if you have single nucleotide polymorphisms or larger mutations in close proximity to these 400 to 430 genes, you have an increased likelihood of developing diabetes. What's also been observed from this initial list of what you would call candidate risk genes is that there's increased incidence of known regulators of beta cell function. So what this has done is really put the focus back on the beta cell of geneticists, of endocrinologists and pancreas biologists as being the beta cell being the primary driver of the pathogenesis and the initial dysfunction that leads to full-blown type 2 diabetes that is observed in the clinic. But when you're working with such large lists of genes, you have to deal with a couple of problems when it comes to understanding the genetic basis. The first is that you need to have an in vivo genetic screening platform to prioritize these candidate genes. And prioritize here, I mean that in a few senses. Firstly, you need to understand which tissues these genes are actually affecting, keeping in mind that type 2 diabetes affects a number of different tissues, of course the pancreas, but also the liver and skeletal muscle. And then also you need to prioritize it in the sense of trying to understand, is it affecting the development of these tissues, or is it later on in the pathogenesis of the disease of type 2 diabetes? And then secondly, in order to really understand how a gene works in a cell type, and in this case in human beta cells, you need to have a method to carry out genetic manipulations in that cell type. And until very recently, there was no method to do these kinds of genetic manipulations in tissues obtained from organ donors. So it was these two problems that I set out to really address at the start of my fellowship at Stanford, and I hope during this talk I can convince you that I was able to take a couple of really positive steps in the direction of trying to address these two issues. So I started off by trying to figure out what is the ideal screening platform, or the ideal genetic screening platform. And I felt that this screening platform needed to really address a few key points. There needed to be a conserved genetic profile. And in terms of what I was looking at to trying to understand regulators of the human beta cell, it needed to be genetically similar to the target tissue, eventually being the human beta cell. There needed to be a conservation of critical genetic pathways. There needed to be a relevant readout or a phenotype, which essentially allows us to understand what effect the genetic manipulation would have on the cell type in terms of that phenotype. And of course, it should ideally be in vivo and scalable, which allows us to work with much larger gene sets. And the system that I found that really ticked all these boxes was the Drosophila fruit fly. Now, in genetics, the fruit fly has been involved in the discovery of a number of critical pathways. More recently, it has been involved in identifying genes that regulate circadian rhythm, and also critical pathways in cell development, such as the Wnt pathway. So over the next few slides, I'll talk to you about the characterization that I did of the Drosophila fruit fly as being the ideal screening platform to identify regulators of human beta cell function. So firstly, what about the conserved genetic profile? So in order to answer the question of how similar is the adult Drosophila IPC, the IPC being the insulin producing cells, which are the orthologs of the mammalian or the human beta cells in the Drosophila fruit fly. So in order to understand how similar these two cell types are genetically, what I did was to purify adult human beta cells from recently deceased organ donors, which I had access to through the Stanford Hospital, carry out RNA sequencing, essentially looking at gene expression across the entire genome, and then do a comparative genetic profile analysis, asking the simple question, how similar is this cell type to this cell type? Just as a side note, when you sequence adult human beta cells, you can see the expression of approximately 22,000 genes, whereas in Drosophila cells and IPCs in particular, you see about 8,500 genes. And both of these are the numbers that have been reported in the literature previously. So how many of these 8,500 genes are expressed, or orthologs expressed in adult human beta cells? What was very, very surprising to myself and others on the project was that 87% of these 8,500 genes are also expressed in human beta cells at a similar level. The remaining 13% can be broken down in two groups. The 11% is the genes that had no known orthologs in human cells, and then 2% were actually not expressed in human beta cells at all. So when you keep in mind that the aim of doing this is to try and identify a cell type that can assess the effect of type 2 diabetes risk genes. So the 450 genes that we're interested in has been identified by GWAS. 99.6% of them fall into this category. So almost all of them are actually expressed in adult Drosophila IPCs. So expression of a gene is one thing, but you need to make sure that there is functional conservation of the critical genetic pathways as well. So what I did following the comparative analysis was a KEGG analysis and a GO analysis, essentially looking at pathways that are conserved and the genes that are conserved across the two cell types. And this is just schematic of a large amount of data, essentially showing you that in mammals and in human cells, particularly, we have insulin, which is packed into vesicles for exocytosis, which is similarly observed in Drosophila IPCs. We have expression of glucose transporters and enzymes required for glucose metabolism in both cell types. There's the expression of a number of ion channels, which is critical for depolarization, as well as calcium entry. And finally, and very importantly, these SNAP proteins. SNAP proteins are a family of proteins that are important for vesicle exocytosis, docking, and endocytosis. All of them are expressed in a similar fashion in Drosophila IPCs when compared to human beta cells. And one of my colleagues published a paper quite a while ago now, just doing a very initial genetic assessment or functional assessment of these critical genes. And what my colleague did was essentially to knock out ILP2, the Drosophila ortholog of insulin, specifically in IPCs, and then try to measure it in circulation. And as you would expect, there's almost complete loss of circulating ILP2, the Drosophila ortholog of insulin, in the flies when you knock down ILP2. And that is accompanied by severe developmental effects. And that's interestingly similarly to what you would see in mice when you knock down insulin. And similarly, when he knocked down GLUT1, the primary glucose transporter, there was also a decrease in circulating ILP2 levels, essentially because the IPC cannot detect glucose, cannot take up glucose, and hence cannot secrete any insulin. So that essentially the take home from this slide is that there is wonderful functional conservation of critical genetic pathways as well. And finally, the critical phenotype. I've already shown you this graph on the right of in vivo glucose-stimulated insulin secretion from a group of mice. And what I found was I can replicate the exactly same thing in flies, where if I fast flies overnight, I can see low levels of ILP2. And if I feed them the next morning with glucose, I see a rapid increase in ILP2 levels in circulation and a gradual decline over the next 90 minutes. So similar glucose-stimulated insulin secretion in the fly, as you would see in mice as well as in other mammals. I want to point out something. ILP2 here is labeled with an HF. That just indicates the HA and the flag tag. Essentially, I epitope labeled insulin here. So I developed an in-house ELISA, which allowed me to measure ILP2 from single flies with a very high degree of sensitivity. So then just to summarize, I hope I've convinced you that this very unique genetic screening platform ticks these critical boxes of having functional genetic conservation of critical genetic pathways and a relevant phenotype, which really allows us to use this as a potential platform for screening type 2 diabetes risk genes. But then the real test for this system would be to push this through a platform where we have genes that have been identified by GWAS and to understand if this system can predict genes that could regulate human beta cell function. So I'm going to show you an initial screen that I did from a cohort of 14 type 2 diabetes risk genes. So all of these genes have been identified by GWAS as being associated with an increased risk of developing type 2 diabetes. And on the left-hand column are essentially the Drosophila orthologs of those genes, which of course are expressed in IPCs. And what I did in this initial screen was to cross these Drosophila lines with RNAi lines, essentially to knock down each of these genes specifically in the IPCs, and then measure the effect on circulating ILP2 levels. And what I observed when we did this initial screen was that there were two lead hits, CG9650 and Pseudo, which are the Drosophila orthologs of human BCL11A and PRC1. So when I knock down each one of those genes independently in the IPCs, it results in a significant increase in circulating ILP2HF levels in the adult fly. So over the next few slides, we'll talk about BCL11A, and then later on, we'll talk about PRC1 as a very unique drug target in human beta cells. So BCL11A is a repressive transcription factor. It's part of a larger transcriptional complex which binds to the promoter region of genes and is able to suppress the expression of its target gene. It has a critical role in regulating the switch from fetal to adult hemoglobin expression in erythrocytes. And because of that role, it has also been put through gene therapy trials currently being carried out at Harvard University for the treatment of disorders associated with erythrocytes such as beta thalassemia and sickle cell anemia. But it has some very interesting observations when it comes to type 2 diabetes. Now, a number of GWAS assessments in ethnic groups across the world has shown that SNPs, or mutations at the BCL11A locus, are associated with an increased incidence of developing type 2 diabetes and associated phenotypes. What that means is that manipulating BCL11A expression potentially may have an effect on beta cell function and hence may increase the incidence of an individual developing type 2 diabetes. But then the question then is BCL11A the key driver? Do changes in BCL11A expression itself regulate the primary role of the beta cell, which is secreting human insulin? In order to answer that question, I collaborated with some of my colleagues who published a paper in PNS in 2014. And what I did was to look at their data specifically in three interesting subgroups. So what they had in this particular data set was islets isolated from non-diabetic donors and type 2 diabetic donors. And they were able to look at three critical markers of stimulation index, serum HbA1c levels from the patients, and by doing RNA sequencing, the expression of a number of genes. But today I'll just show you the expression of BCL11A. So stimulation index is essentially an assessment of how healthy an islet is in terms of how much insulin you can secrete. So as you would expect, non-diabetic individuals have a high stimulation index because they're able to respond to glucose by secreting insulin. Type 2 diabetics, on the other hand, can be divided into two distinct groups in this particular data set. You have glucose responsive type 2 diabetics. So these are individuals that have maintained some amount of insulin secretion from their pancreatic islets. And on the other hand, you have this final group of type 2 diabetics, which we've labeled as glucose unresponsive. So they've almost completely lost their ability to respond to glucose by secreting insulin. Unsurprisingly, this group that cannot secrete insulin from their pancreatic islets have the highest serum HbA1c levels. As I'm sure you're familiar with, HbA1c levels essentially is an assessment of your long-term glucose regulation, where high HbA1c levels is indicated of hyper and in certain cases hypoglycemic episodes as well. And as a control, of course, your non-diabetics with a high stimulation index have that lower HbA1c level, essentially showing you they have tight or better glucose regulation. So the question then was, what is the BCL11A expression across these three groups, specifically in the pancreatic islet? What was very excitingly observed was that in this group that had the lowest ability to secrete insulin, there was the highest islet expression of BCL11A at an mRNA level. So this really leads to this hypothesis that more BCL11A in the pancreatic islet and most likely in the pancreatic beta cell is a detrimental thing to the cell, which then inhibits insulin secretion. So then the question was to ask directly is, does BCL11A actually regulate beta cell function? But in order to answer that question, I needed to develop a system to do genetic manipulation in human islets. So the method that I developed was the human pseudo-islet organoid culture system. And in this system, I start with human islets that I obtained from recently deceased organ donors. And after a lot of optimization, the method that I've selected was to dissociate these clumps of human islets. Just a reminder, that's approximately 1,000 cells in each human islet. They're dispersed into a single cell population, at which time I introduce lentiviruses carrying vectors to knock down or overexpress any gene of interest. And I can then culture them under very specific conditions for 3 to 4 days and force their reaggregation into these 3D spheres or organoids, which we refer to as pseudo-islets. This is just a bright field image showing you a group of these pseudo-islets. Each one of these, for example, contain about 800 to 1,000 cells. And as you often do when you're working with lentiviruses and vectors, you have a fluorescent reporter to make sure that you're transducing or efficiently genetically manipulating these cells. And when you look at the GFP expression of this same image, it's hard to tell from this image, but approximately 80 to 90% of these cells are GFP positive, essentially indicating to us that there's a high level of viral transduction, which should translate into efficient gene knockdown or overexpression, depending on the type of vector you're using. After 3 to 4 days, what I did was to collect some of these organoids and just stain them with antibodies against insulin and glucagon, and then visualize them under a confocal microscope. And what you can see here is after 4 to 5 days, they still express those key hormones. So insulin and glucagon are expressed throughout those cells, confirming that they still produce those two critical hormones, which is important for the function of these cells. Functionally, these pseudo islets behave very similar to what we see with human islets in vitro. So what I've done in this particular experiment is an in vitro human insulin secretion assay, where I took a group of 20 of these organoids and I cultured them at low glucose for 1 hour, and I measured the amount of insulin secreted into the culture media. I can then transfer those same organoids for 1 hour at the high glucose concentration. And what I observe, and I've consistently observed, is that there's a significant increase in the amount of insulin that's secreted into the culture media, essentially confirming that these organoids are still healthy and they maintain their key function of glucose-stimulated insulin secretion. So unlike the previous data I showed you in mice, which was done in vivo, now this is an in vitro method to assess glucose-stimulated insulin secretion. One of the key things that was holding back development in working with human islets was that once you isolate human islets from human donors, is that you can keep them in culture for 4 to 5 days, after which time they become necrotic and they undergo rapid apoptosis and they die. So I wanted to develop a method that allowed me to do experiments with these organoids for extended periods of time, especially given that I'm doing genetic manipulations and I want to understand the long-term effects of those genetic manipulations. So the method that I developed required me to transplant these pseudo-islet organoids under the kidney capsule of immunocompromised mice. So immunocompromised means that they would not reject these organoids over time. And then what I was able to do was to actually measure human insulin circulating within the mouse. So this is some version of a humanized mouse that I was able to generate. So 6 months post-transplantation of these organoids, what I did was to inject glucose into the recipient mouse and then measure human insulin in the serum. And what I was able to see 6 months post is that these organoids are still healthy and functional, as when I inject glucose, those organoids respond by secreting more human insulin in a very regulated fashion. I've since done experiments that have been able to keep these organoids alive for 12 to 18 months. So really extended the long-term possibilities of doing experiments with these genetically manipulated organoids. And of course, you can do some very interesting histology or microscopy by harvesting this kidney. And this is a representative image of one human pseudo-islet, which is sitting under the kidney capsule and on the kidney. This is just an H&E image, but you can do a lot of very interesting confocal microscopy to look at the expression of critical markers that you may be interested in. So I use this human pseudo-islet system then to ask a very simple question. If I overexpress BCL11A in human pancreatic islets, what effect does that have on insulin secretion? So I used a construct delivered by lentivirus to overexpress BCL11A, and I carried out an in vitro glucose-stimulated insulin secretion assay. I'll explain this data a little bit carefully because there's a few versions of this over the next few slides. Essentially, what you want to look at in these experiments is in the control samples, which is essentially infected with this control lentivirus, is when you culture the organoids at low glucose, you expect to see some amount of insulin being secreted, but then you expect to see a stepwise increase in insulin secretion when those same organoids are transferred to high glucose, or 16.7 millimolar, or 16.7 millimolar plus IBMX, which artificially depolarizes the cell. So this stepwise increase in the control confirms that your organoids are healthy, they're alive, and they still maintain that key function of glucose-stimulated insulin secretion. Once you confirm your control is working, you can then look at the effect of the BCL11A overexpression. And you can appreciate here that overexpressing BCL11A completely blocks the ability of this cell to respond to high glucose by secreting insulin. And this becomes more obvious when you try to artificially depolarize with IBMX. So essentially, in line with the observation from the clinical samples that I showed you previously, where more BCL11A resulted in less insulin secretion from the beta cells. So then we need to dive into the mechanism. How does BCL11A expression actually regulate human insulin secretion? And given that BCL11A is a repressive transcription factor, I wanted to do the opposite of what I just showed you. I wanted to now knock down BCL11A, form those pseudo islets, purify the beta cell, and then carry out RNA sequencing. Essentially, I was looking for the expression of genes that were going to go up as markers that were downstream of BCL11A, trying to understand the downstream targets of BCL11A, which may lead to changes in insulin secretion. This is, I just put these two representative FACS plots in to show you that I'm able to specifically select GFP positive. So in this case, lentivirally transduced cells. And then by staining those GFP positive cells with glucagon or insulin, I can specifically select out either beta or alpha cells. And in the analysis that I'm going to show you today, because we're of course interested in insulin secretion, we are going to talk about the beta cells specifically. So there's many types of analysis you can do with RNA sequencing, as I'm sure you're very familiar with. One of them, of course, is gene ontology, which simply put is looking at pathways that are changing. So what I did was to look at pathways that were upregulated following knockdown of BCL11A. And very interestingly, I found that when you have less BCL11A, the pathways associated or the genes associated with insulin secretion, endocytosis, or microtubule-based processes were all upregulated. So if you look at the specific genes that are driving these pathways, you can see that there's some very, very interesting genes here. For example, there's transporters, potassium channels, and a number of SNAP proteins. It has been shown in the literature that upregulation of each one of these genes individually is sufficient to enhance insulin secretion. So it appears that BCL11A is an upstream regulator of a number of genes here. And we've done some further analysis that I won't talk about today to show how it regulates the expression of some of these key genes. So reducing BCL11A gives us this very interesting genetic signature that suggests to us that it could enhance insulin secretion. But I wanted to prove that. So I went back to my pseudo islets, and then now we have the opposite of where I've knocked down BCL11A, and that resulted in a significant enhancement of insulin secretion as predicted in the prior slide by the RNA sequencing. So the organoids that had led BCL11A had higher insulin expression, secretion at low glucose, as well as at high glucose, and this interesting trend when you artificially depolarize the organoids as well. This experiment was done 4 days after transduction, because this was done in vitro, and that's about as long as you can do those experiments with this tissue. So I wanted to ask if this improvement in insulin secretion persists for extended periods of time, and the answer is yes, it does. So 2 months post-transplantation, I injected glucose into recipient mice that had either control pseudo islets or pseudo islets that had BCL11A knockdown. You can see that enhancement of insulin secretion persists for extended periods of time. Again, this experiment was done 2 months post-transplantation, but I've observed this enhanced insulin secretion for 6 months post as well, really sort of suggesting that we've completely reprogrammed these cells. By just removing one transcription factor, you can direct these beta cells to secrete much more insulin, which is what you would hope to see in type 2 diabetic patients that have less beta cell mass. So just to summarize that part of the talk then, I've shown you that I've developed 2 interesting systems. Firstly, an in vivo screening platform in the Drosophila fruit fly, which is a predictor of genes that could be affecting human beta cells and more specifically insulin secretion. I then developed a human organoid system amenable to genetic manipulation and long-term assessment. So this was the first time that we were able to do genetic manipulation in human tissue. And then using a combination of these two systems, I was able to identify BCL11A, which is a repressive transcription factor and a novel regulator of human insulin secretion, and really sort of leads us into a further understanding of why this has been identified by GWAS as a potential risk gene when it comes to type 2 diabetes. So I really wanted to, having sort of really confirmed that this system works, I wanted to push the system a little bit further and see if I can use it now to discover novel therapeutics, particularly for the treatment of type 2 diabetes. And in my second candidate there, Pseudo, which is the Drosophila ortholog of human PRC1, I felt that I had a very quote unquote druggable target. And the reason I felt that was because PRC1 is referred to as a protein regulator of cytokinesis, and it is a microtubule associated protein. So in this schematic, you can see that this is a microtubule where the two subunits of beta and alpha tubulin are shown. PRC1, like I mentioned, is microtubule associated. It is very much like an arm that reaches out from the microtubules. And its function is essentially to capture other microtubules in an antiparallel fashion. So this is minus to plus, whereas this is plus to minus. So given that its role is to really hold microtubules in place and essentially affect microtubule structure or morphology, it is unsurprising then that if you affect PRC1 expression, you affect microtubule morphology. So if you overexpress PRC1, essentially if you have more of these connections between different microtubules, you get extensive microtubule bundles or much more rigid microtubules or more tightly packed. And then conversely, if you knock down PRC1 or have less of these arms, you get a much less dense microtubule meshwork. And this is particularly critical during cytokinesis, the last stage of mitosis. So if you knock down PRC1, cells cannot undergo cytokinesis and you have binucleated cells because there is improper separation of the two daughter cells. But in human beta cells, microtubules have a very, very interesting role. It has been shown in a number of papers, I've just given one reference here, that shows that microtubules negatively regulate insulin secretion and vesicle mobility. So if you look at the microtubule meshwork in a human beta cell, it is very dense, it is highly disorganized. And when you have high glucose, you see an unpacking of that meshwork, which allows vesicles to move towards the membrane and undergo exocytosis. So that microtubule meshwork is very dynamic. In its resting state, it's highly packed, essentially preventing the insulin granules from moving towards the membrane and undergoing exocytosis. So the first question then is, given that we know the role of microtubules and the role PRC1 plays in maintaining microtubule morphology, is to ask if PRC1 itself can then regulate human insulin secretion. So I went back to my trusted organoid system. I developed a construct to be able to knock down PRC1 expression by a little bit over 50%. And I then carried out an in vitro glucose-stimulated insulin secretion assay on these organoids that had less PRC1 expression. And I'm hoping you're pretty familiar with these kinds of data now, but essentially just a quick summary is that when you knock down PRC1 expression, you have enhanced insulin secretion. And this is true at low glucose and so more obvious at high glucose. So essentially less PRC1 translates into more insulin and essentially enhancing the function of the human beta cell. So given that we know if we genetically reduce or genetically knock down PRC1 expression, I wanted to see if I can identify a small molecule or families of small molecules that can similarly pharmacologically inhibit PRC1 and then potentially improve human insulin secretion. So in order to do this, I designed a 3-tier small molecule screening platform with the aim of identifying compounds which suppress PRC1. So the compound selection I did here, it was not a high throughput, it was just 20 to 30 compounds. It was a highly selective compound library, which I selected via literature searches, some in silico modeling, and then some of the compounds were synthesized in collaboration with some chemists at Stanford University. So in the first year, I was going to use the Drosophila fruit fly, and I was essentially going to feed these compounds to the fly and assess if there were improvements in ILP2HF secretion. Any compounds that improved ILP2HF secretion would move on to the second tier, where I would use human islets from organ donors. The lead compounds would then be used for an in vitro treatment, and I would assess insulin secretion. And now I want to become a little bit more specific and understand if those improvements in insulin secretion were actually signaling through changes in PRC1 expression. And then the lead molecule from tier 2 would go into a more in-depth analysis, of course, mechanism of action, some safety studies, and in vivo studies such as pharmacokinetics and safety to see if this has potential to move forward potentially into clinical trials. So the compound that I identified from this 3-tier screening platform, we refer to as B303. Now, this is a naturally occurring amide alkaloid small molecule. It has been shown in some publications to have potent anti-cancer activity in a number of different cell lines. And in certain cases, so in pancreatic, gastric, and colon cancers, it has been found to reduce PRC1 levels as well. I can't talk too much about this compound in terms of the chemistry or the structure, as it is patent protected right now, and forms part of a larger patent or a family of compounds for enhancing human insulin secretion. But I can talk about the data, and I'll talk about the 3 tiers over the next few slides. So starting with the fly then, what I did was to feed B303 to flies for 3 days. And at the end of 3 days, assess the effects of the compound or the vehicle on fasted as well as fed ILP2HF levels. And what I found was that B303 had a dose-dependent effect on secreted ILP2HF levels. So I've done 2 things here. I've fasted the flies overnight and then fed them with glucose. In the controls or the vehicle controls, you see a stepwise increase in insulin. And that stepwise increase becomes much greater as the concentration of B303 increases over that 3-day period. So given I found this profound increase in ILP2HF levels in the fly, I next wanted to move this compound into the second tier where I wanted to work with human islets. So I treated human islets with B303 for 48 hours. You can see that there was some optimization that was required in the timing when you move between the fly and the human islets. And this is pretty consistently seen with a lot of other compounds as well. So after 48 hours, I found that B303 suppressed PRC1 expression to a similar level to what I was able to achieve genetically in human islets. So this is just a qPCR that I carried out, essentially assessing PRC1 gene expression in the whole islet. So what effect does this suppression of pharmacologically of PRC1 have on insulin secretion? And very excitedly, across the board, there was a profound enhancement of insulin secretion. And this was true at low glucose as well as high glucose and high glucose plus that depolarizing agent. I just want to point out this data is generated from 5 human islet donors, but subsequently this experiment has been carried out in approximately 15 to 20 donors. And this enhancement of insulin secretion is true independent of age, sex, BMI, and in certain cases, type 2 diabetes status as well, where in all cases, treating with B303 enhances insulin secretion from human islets. So just quickly, we'll talk about how B303 enhances or improves the human insulin secretion from the beta cells. Firstly, we did some patch clamping in collaboration with Dr. Herbert Gaisano at the University of Toronto, who's an expert in patch clamping human beta cells. And just a quick summary, what he did was to measure capacitance, where capacitance essentially measures how many vesicles are undergoing exocytosis as a function of the surface area of the cell membrane. So what Dr. Gaisano and his postdocs can do is artificially depolarize a beta cell, forcing it to undergo exocytosis and then measure capacitance. And hence, he can tell you how much exocytosis is happening. So all of these experiments are paired. And what that means is that we divided these beta cells from the same donor to be either treated with the vehicular control or B303. So the vehicle control first, when you undergo these depolarization, or the 10 pulses as it's referred to, you see a gradual increase in cumulative capacitance. Essentially, confirming that the beta cells are undergoing exocytosis because they are being artificially depolarized. But what happens when you pre-treat with B303 is that there is a significant enhancement in capacitance. Essentially, there is much more exocytosis that's going on over this period of time. It's almost a 3-fold increase in exocytosis. So really in line with what I've shown you previously, that there's more insulin detected in the culture media when you treat with B303, and that is due to almost 3-fold more exocytosis occurring. So this also suggests that the vesicles are getting to the membrane more efficiently. There's much more vesicles at the membrane. So how does B303 actually improve this vesicle mobility? And in order to answer that question, we had to go back to the microtubules, of course. In order to really look at what effect B303 has on the microtubule meshwork, I carried out some super resolution microscopy in primary human islets. So what you're looking at here is essentially the edge of a human islet stained with alpha tubulin and insulin. So you can see, as I mentioned in a few slides ago, that the microtubule meshwork of a human islet is very dense, it's highly disorganized, and essentially it is evenly dispersed throughout the cytosol of the cell. Similarly, insulin, when you stay with insulin, you can see these insulin granules essentially marking the individual insulin vesicles that are also dispersed uniformly throughout the beta cell. In this field of view, I just want to point out there are 4 beta cells that you're looking at. There's 1 here, 2, 3, and 4. And then the question is, if you pre-treat with B303 and you did this kind of microscopy, would you see changes in microtubule density? So that's exactly the experiment. So I've already shown you this figure. Essentially, just want to point out again that the insulin punctae and the insulin vesicles are dispersed throughout the cytosol. But when you pre-treat with B303, you see how all of those insulin vesicles are now in close proximity to the membrane, so much so that there's hardly anything within the cytosol. And that's true of this beta cell here and this one here as well. And the reason that all of these vesicles are in close proximity to the membrane is because there's a complete unpacking of the microtubule meshwork or the microtubule morphology is essentially completely changed. In the controls, you can see how it's dense and highly packed, whereas if you treat with B303, it is unpacked. There's much more spaces in there as well. And that's just the merge, once again, confirming that this is indeed a beta cell, as are those 4 cells there. With a computational biologist that I collaborated with, was able to draw up an algorithm that was able to calculate the distance between microtubules in random locations across the beta cell. And what this data shows you is that there is a consistent increase in the distance between microtubules when you pre-treat beta cells with B303 when compared to the vehicular controls. So really confirming to us that, the movement of vesicles close to the membrane is indeed due to this unpacking of the microtubule meshwork. So in my last data slide, I just want to show you some data that confirms that all of this in vitro data that I generated with B303 translates into an in vivo phenotype. So this is a bit of preclinical data using mice where I injected B303 once daily for 5 days at 2 different concentrations. And at the end of that treatment regime, I did a glucose-stimulated insulin secretion assay. And the data was very promising and very exciting. It showed that at the higher concentration of B303, so 2 milligrams per kg, that there was an enhancement of fasting insulin levels. And then, of course, when you inject glucose and look at the insulin excursion over the next 2 to 3 hours, there is significant enhancements of insulin at the higher concentration of B303. But there was, of course, a suboptimal dose, which was very similar to the vehicle control. So a very dose-dependent effect of this compound, which is what you would expect to see and hope to see if you're having very specific effects of this molecule. So in summary then, I've shown you that I'm able to now target genetic variants essentially by identifying a potent inhibitor of PRC1, which is B303. It is capable of enhancing human insulin secretion. B303 alters microtubule dynamics in a very time and concentration dependent manner. I just showed you a snapshot at 48 hours, but you can do much more sort of fine-tuned experiments where you can see the microtubule meshwork changing over time. But at 48 hours is essentially where you really see that profound unpacking of the microtubule meshwork. There's a lot of ongoing preclinical work for patents and also for some publications that are close to coming out now, looking at the efficacy of B303 in human type 2 diabetic islets. We're also trying to optimize the drug delivery methods with the aim of trying to minimize the potential off-target effects. And they also have a big medicinal chemistry effort, really trying to improve efficacy and potency, and also improve the in vivo half-life of this small molecule to give it a better chance if and when it moves into the clinic. So in conclusion then, I've shown you the power of functional genomics, essentially. We can start with all these wonderful GWAS studies that have been carried out in multiple groups. I've spoken about type 2 diabetes today, but I think this is true across multiple disease spaces. GWAS gives us long candidate list genes, but by developing screening platforms in unique systems, you can really understand how these risk genes contribute to the pathogenesis of a disease. In the case of BCL11A, I identified a novel regulator of insulin secretion, and by doing some in-depth mechanism of action studies, we really know how this transcription factor regulates a number of downstream targets. And then, of course, in the case of PRC1, we now have a very novel, unique, and a new target, which we feel has tremendous potential for enhancing beta cell function and maybe a potential treatment for type 2 diabetic patients. So with that, I just want to acknowledge a few people. Firstly, the organ donors and their families for their very generous contributions that allow us to do these very interesting experiments in human tissue. The members of my previous lab at Stanford, collaborators across the world, and of course the funding bodies, specifically the American Diabetes Association and the Child Health Research Institute at Stanford. And with that, I'm happy to take any questions. I also have my email address here. If you have any further questions or you want access to any of these sequencing data sets, I'm more than happy to discuss that and share that with you. So with that, I want to thank you for your attention, and I'm happy to take any questions if there are any. Thank you.
Dr. Heshan, thank you so much. It was a very comprehensive presentation, and I congratulate on the amazing work that you have been doing. To start off the question and answer session, I would like to get your opinion on organoid cultures and what do you think about stem cell transplantation, especially the Edmonton protocol that some centers are using. Is it of any use, or do you think that's just bypassing some of these pathways and directly going into like clinical trials? What are your opinions on it?
I think the stem cells, especially sort of the stem cell protocols that are now being used, there's some being developed at UCSF. I think the initial versions of the stem cells that were being generated were what they referred to as beta-like cells. They were not complete beta cells. They lacked some of those key factors of being glucose responsive and secreting insulin. But I think there's a next generation of stem cells and that are much more like beta cells. Some of those are being generated at UCSF, I know, where the beta cells are much more similar to an adult beta cell. Essentially, they have much better regulation, much better signaling from glucose to insulin. So I think, I mean, to answer your question, I think the stem cells have tremendous potential, and I think the way they are being developed now are much more similar to human beta cells. So I think the Edmonton protocol and the newer transplant protocols have tremendous potential for treating the loss of beta cell function and returning glucose homeostasis. The only issue though is the number of cells that you need to infuse into these patients, I think is still limiting. There's a large amount of cells that need to be infused in order for the transplant to actually, for the patient to actually become insulin independent for extended periods of time.
Okay, thanks. Any other questions? Okay, so in the absence of any more questions, I think we can conclude this meeting, and the recorded version of this webinar will be linked, and I will send you the links personally. Thank you so much. And thank you, Dr. Heshan, for taking your time to prepare this presentation and explaining everything very in detail, and it was very clear for us. Thank you so much.
Thank you. Thank you for having me there.